Good morning. So, I'm going to take on the topic models of DNA replication. So, models of DNA replication, we have already studied about the replication, the process, how it occurs. So, here I'm saying the models of DNA replication. So, as an intro, we can see that the model of DNA replication suggests that two strands of the double helix separates during replication, and each strand serves as a template from which the new complementary strand is copied. That we knows that what is the process of DNA replication, that there will be a double helix. It will unwind during the replication and it becomes the template stand, and according to the template stand in corresponds to that, it makes a complementary stand that we know it. So that's the just an intro type it's said, and we are going to learn about three types, that is rolling circle model, that means three models, that is rolling circle model, d loop model and theta model. So starting is first one, rolling circle model. In rolling circle model, we can see that it's mainly in prokaryotes, that is especially in bacteria we can see this type of model, which possess the plasmids. A unique type of replication occurs known as rolling circle replication. And the specialty of this rolling circle model is it is unidirectional. Unidirectional means just one way steps or one way formation it is, and this can rapidly synthesize the multiple copies of circular molecules of DNA or RNA. So it can multiply the copies or synthesis. It can rapidly multiply the circular DNA and RNA's copies. And in this rolling circle model, we can divide it, or we can say it in three steps. That is, as we said before in replication, the steps, same way here also there are three steps. That is initiation, elongation and termination. Okay, so we said the three steps: initiation, elongation and termination. Now, first one is initiation. Initiation, we can see that it's a nick one of the two strands producing free 3 OH and 5 phosphate ends by helicase topoisomerase single strand binding protein. That means you can see in the picture, that first picture which is shown is the bacterial plasmid, the proper way of bacterial plasmid. And at the time of initiation process, what happens is nick1. Nick means the small cut occurs. You can see in the second diagram, that's that. In the second diagram, you can see here there is a shortcut happened. Cut had been occurred. So that cut occurs, that is said as that nick one. That means nick means a small cut. So a small cut occurs there, and the outer stand, this outer stand, it becomes one end has become 3 dash OH. I mean in 3 dash OH, and at 5 dash there was phosphate group. So it how it occurs is it occurs with the help of helicase topoisomerase single stranded binding protein. With its help, the initiation process occurs. So what happens in initiation? mm, so what happens in initiation process is nothing but a small cut occurs. Uh, Small cut occurs with the help of the enzymes helicase, topoisomerase, single strand binding proteins, and at 3 dash end there will be OH group, and 5 dash there will be phosphate group. That is the initiation process or initiation stage. So that was the first step of initiation stage. Next is elongation. So yeah, next is our elongation step. In elongation step, what is said is the OH group of broken strand with the unbroken strand has a template. The polymerase will start to move in a circle for elongation, due to which it is named as rolling circle model. Then 5 dash end will be displaced and depart similarly to a thread. So yeah, in this process, what it is said is that OH group of broken strand with unbroken strand has a template. That means uh, you can see here that OH strand with this template strand. the this second round, this template OH strand. With this template strand, that is of an unbroken strand, with the help of this DNA polymerase 3, we have studied about the DNA polymerase 3, what are its importance. So with the help of it, it starts to move in a circle for elongation, that you can see here. You can see this point was first present here, then from there, first present here, from there it started to elongate. Why elongating in a circle motion with the help of polymerase enzyme? And at the same time, it occurs in 3 dash, and at the same time in 5 dash end, what happens is the strand which is there, it start to displace from here. You can see that it starts to displace from there. Displace means it starts to move from its original position to a different position. So that is the process occurring here. First we have said there was a cut made where 3 dash and OH was present and 5 dash and phosphate was present. And in second process we said that with the help of DNA polymerase 3, the portion of 3 dash and started to elongate and in a circular motion. So it is known as rolling circle model because it is rolling in a circular way. And at the same time, the 5 dash end or the 5 prime, that end moves to displaced and depart similar to like a thread type. It started to displace. Displace means it moves from its original position. That was occurring here. So that is the process of elongation. Now the last process is termination. Termination, what happens is at the end of the termination, the linear DNA molecule is cleaved from the circle, resulting in a double-stranded circular DNA model and a single-stranded linear DNA model. It's simple. Here we can see when the cleavage occurs. Here you can see the cleavage occurs here, and after the cleavage occurs there, it moves to a different strand and it forms a double-stranded DNA circular molecule and a single-stranded DNA circle molecule. Just simply it is said that after the rolling process occurs, that is we have said in elongation, how it elongates, how it circulates. After that the cleavage occurs there, and the origin you can see after the elongation process it have moved to this place. First it was present here, from there after elongation process it had moved to the place. So after completing it, it had moved to, it had made two ways, that is one, it had formed to a double-stranded DNA circular model and a single-stranded DNA model. And the linear single-stranded molecule is circularized by the action of ligase. So this is done, that is a linear single-stranded molecule is circularized by the action of ligase, with the help of ligase, and then it is replication to double-stranded circular plasmid molecule. So what is said was rolling circle model. There are 3 steps: initiation, elongation, and termination. In initiation, with the help of the enzymes helicase, topoisomerase, or SSPV, they had made a small cut where 3 dash and it has OH and 5 dash has P, and the origin is from 3 dash. And then elongation process with the help of DNA polymerase 3, from the 3 dash the origin, the strand had been broken and it started to elongate in a circular rolling circle model, in a circular way. So it is said as rolling circle model, and at the 5 dash end it started to displace from its original position like a thread. And at last it is termination. In termination process, what happened? A cleavage occurred and the original position had come to an end, that is at 5 dash, and then it was moved and it formed a linear single stranded molecules, and with the help of the action ligase, and then it became replicated and formed double circular plasmid molecule. It is a process occurred. In this picture I have shown, as I said before, the process of initiation, elongation and termination. All these diagrams together it is seen. You can see here, first it was a bacterial plasmid where helicase and it was present. Then the strand was broken down and the DNA polymerase came here, and then so that was the first process, we said initiation process. Now next is a elongation process. There you can see the 3' and had been elongated from its original position in a rolling model, rolling circle model, and at 5' end it started to displace from its position, and then the cleavage occurred, and then cleavage occurred and 5' and you can see the thread like one half came outside, and then with the help of ligase that 5' end had became a linear, I mean a single stranded DNA, and then after the replication it had become to double stranded DNA. So this is a process of a rolling circular model. It's also same thing, it has been shown in a downwards model. And then the example for this rolling circular model, we can say the conjugation between F plus and F minus bacteria. This for showing how it is moving. You can see in first figure, it's just to show that how it is occurring, that's all, nothing more. Here you can see how the first one it was kept. There was a F factor present, that means the plasmid which is present, and then there becomes a cut had been occurred between them, and then with the help of elongation process the 5' and had been moving, then it moved and it was started to circle, then at last the two of them had been made. It is simple, that the process which we said before, same it is here too, nothing is different in that you can see. For just to understand how it is occurring, I have just put this picture. So yeah, that's all. So that was all about our rolling circle DNA model. Now we are going to say about D-loop replication. First it was rolling circle replication, next is D-loop replication we are going to see. So D-loop replication, if we see, it is the process by which circular DNA like chloroplast and mitochondria replicate their genetic material. So it can be seen in chloroplast and mitochondria that how it replicates its genetic material. And here the D-loop was first discovered in 1971 when researchers noticed that many DNA in a mitochondria, they were examined under a microscope containing a short segment that was triple stranded. So while they were having an examination in the mitochondrial, the DNA present in that, they have seen the D-loop first. And the important compound of D-loop is that many chloroplasts and mitochondria have a single circular chromosome. Like bacteria, we said bacteria is having a single circular chromosome instead of a linear circular chromosome which can be seen in a eukaryotes. It's just an information, just what is it, that's all. And in many organisms, one strand of DNA is the plasmid compares heavier nucleotide, that is purines, adenine and guanine, and this strand is called heavy strand. So yeah, for this process there is two strands, that is heavy strand and light strand. Heavy strand means it will be of purine, that is adenine or guanine will be present, so it is said to be heavy strand. And if it's light strand, we say it has present lighter nucleotides will be present, that is pyrimidine, thymine and cystine. You can see here H is written, that is inner part is the heavy strand and outer part is the light strand. You can see there, for showing that I have been put this. Then the replication begins with replication of the heavy strand starting at the D loop. So the replication here occurs in the heavy strand, starting from heavy strand. Then a short portion in a circular DNA has 3 strands instead of 3, and the D loop specialty is that we said almost we will be having 2 strands, but for the D loop strand there will be 3 strands instead of 2. and the middle strand will be the complementary to the light strand displacement the heavy strand and forms a displacement loop. And the circular DNA is stable with this D loop. So when we compare, it's before we seen circular DNA and D loop, the circular DNA will be more stable and can remain in the formation. But the middle strand of the displacing strand is replaced frequently due to its short life and is very energetically expensive to the cell. So this middle portion, we said there are three strands. So the middle strand, it will be short lived. And if we look at this, and when the diagram resulting structure looks like D, we are saying that it will be D loop. It's not the complete diagram which I am showing, it's just to show that how it is. That you can see light band and heavy band inside and light band outside. Then with the initiation at the origin of H, you can see a DNA primer reaches there, right? Then the synthesis of DNA occurs. That is, H has a template and it works, and the DNA occurs. So when the RNA primer comes there and when it synthesizes, you can see a D type process. You can see something like this curves occurring there. So yeah, newly synthesized strand displaces L strand. So see how when it occurs, here you can see RNA primers came, then the synthesis occurred in DNA, and H, with the help of that H strand, has a template strand. It made a complementary strand, due to which there occurs a loop, and that loop was of D shape. So we said that it is D loop or D loop structure. We say it has D loop structure. So it's just an information that in D loop what is present. That is light strand is present, heavy strand is present. Heavy strand means it will be purines, and if it's light strand it will be pyrimidines. And when the initiation occurs firstly at heavy strand where RNA primer comes, then the synthesis of DNA occurs. That time the H strand will be working as a template strand, and then when it correspondingly forms a new strand, then it forms like a D shape. So we can say it has a D loop. Now we can see how the replication process occurs. That is, each D loop contains an origin of replication for a heavy strand. We have said before that heavy strand is present. The full circular DNA replication is initiated at the origin and replicates in only one direction. We have seen here it replicates only at one direction. Yeah. And the middle strand in the D loop can be removed and a new one will be synthesized that is not terminated until the heavy strand is fully replicated, or the middle strand can serve as a primer for the heavy strand replication. We have said before that there will be three strands, right? So first strand we can see heavy strand, then we can see outer light strand, and that RNA primers when came here and formed, we can say it has third strand, that is the middle strand. And as a heavy strand replicate reaches the origin of replication of the light strand, a new light strand will be synthesized in the opposite direction as the heavy strand. Like heavy strand and light strand will be also formed while the primer had been kept. So that was about the replication process, how it occurs. As we said before, it's like same, that the first initiation occurs in the X strand, and then it moves. Initiation occurs in the H strand, then it moves, then a primer forms, and then it the primer works as in middle portion, that is it works as an when H becomes the template strand, the primer, that portion starts to elongate and it works as a newly synthesized form for the light strand. According to light strand, it will also be changed. That means in heavy strand is present there. When the heavy strands become template strand, according to the template and corresponding to that, a strand will be formed which will be similar to the light strand, and it will be forming in a circular mod way, or in a way that it will look like D shape. So we can say that it is a D loop model. So that is shown here. Now next is what is the importance of D loop. That is, the D loop region is important for phylogeographic studies. For phylogeographical studies, the D loop is used because this region do not code for any genes, and it is imperative for this region to remain conserved over time. Therefore, it is free to mutate with only a few selective limitations on size and the mutation rate is among the fastest of anywhere in either the nucleus. That means in this do loop, the mutation rate will be more. And using that mutations in the D loop, we can see the evolutionary changes taking place. And it is very common in D loop in genomics. So that was about the D loop model. Now last is our theta model of replication. Theta model of replication, we can say, it's a common type of replication that takes place in a circular DNA. That is such as like E. coli bacteria. It is taking place in E. coli, this theta model, and other bacteria, which is called theta replication. Why we are calling it as theta replication is because it generates a structure resembling to Greek letter theta. We know how, so we know that how we are writing theta, right? We used to write the Greek word theta like this. So has the process of theta or has the replication occurs here, it resembles like a Greek word like theta, so we can say it has a theta replication. Now what occurs there is, in theta replication, double-stranded DNA begins to unwind at the replication origin, producing single-stranded nucleotide strands that then serves as template on which new DNA can be synthesized. In this, all the same process occurring, that is, double-stranded DNA begins to unwind. It unwinds and produces a single strands of nucleotide strand, in which one strand works as a template strand, and corresponding to the template strand, a DNA will be synthesized. Same process is happening here too. And this unwinding the double helical generates a loop term, a replication bubble. So when here the double stranded helical structure unwinds, it forms in a loop structure, it forms in a bubble structure. And unwinding may be at one end or at both ends, and the DNA replication of both the template strand is simultaneously with unwinding. It's nothing but just simply there will be a double stranded DNA, it will be unwinding, and unwinding time there will be forming a single stranded DNA, and that single stranded DNA acts as a template strand, and according to the templates and the corresponding codes will be made. According to that, DNA will be synthesized. It's a simple process, and while the time of unwinding it, there forms a replication bubble shape. So that shape will be of theta type. We can see how it is. We can see the origin of replication there. As the origin of replication, it starts to unwind. Local opening of double helix. We have said the double helix starts to unwind. So how it's unwinding, you can see there, right? And then with the help of replication fork, it unwinds and it forms like a theta shape, and it forms like a theta shape. You can see here it starts to unwind, and while it was unwinding, you can see it is forming in this position, in this way. So that means it is forming in a theta way. That's why it was said as theta formation, and after that, with the help of bidirectional replication, it forms into two new strands. So if there are two replications, now we are saying that about, we have said about how the process happening, and now we are going to say that if there is two replication forks, and if there is one replication fork, what will be happening? So we can say that if there are two replication forks, one at each end of the replication bubble, the fork proceeds outward in both directions in a process called bidirectional replication, simultaneously unwinding and replicating the DNA until they eventually meet. So if two replication fork is present, what happens is one at each end, each replication forks will be present at the both the end of the replication bubbles, and that proceed to outward. I mean, it comes outward. It forms like an outward. It forms to an outward direction, and that process will be called as bidirectional replication. You can see here we have written here is the bidirectional form. So that way the bidirectional form it will be formed, and it will be unwinding at the same time till the replication meets it. And if it is, it's in the case of two replication forks, and if there is a single replication fork present in it, it proceeds around the entire cycle to produce two complete circle DNA molecules, each consisting of one old and one new nucleotide strand. So if it's a single replication fork, what happens is the process of DNA replication occurs twice, in which one will be old strand and another will be a new strand. So this is the difference when single and double two replication forks occurs. So yeah, this was about the theta model. So we have studied now three models, that is rolling circle model, D-loop model and theta model. I hope this is clear to you, because it's not that so tough to understand, because in first we said rolling circle model, we said there is initiation, elongation and termination, where we have seen that in initiation step with the help of enzymes the cut is formed and 3' and 5' is formed. Then in elongation the elongation occurs in the 3' and that is an origin, and from there in the circle model the elongation process occurs, and then the cleavage occurs and the 5' end moves out and it forms a linear structure. Then with that it forms a double structure. And then we said about D loop replication, we said that there is heavy strand, light strand present in them, and we said has heavy strand become the template strand and RNA primase scheme, and it corresponding to heavy strands, that is template strand. According to that, a new strand will be formed, which we can see in the middle portion, which will be complemented to which will be equal same as the light strand, and then that strand when formed, it forms in a D shape, so it can be said to be D loop. We said, then we said about theta model, that how the theta form is happening, and if there is a single fork and double replication fork, what happens, that is if there are two replication forks, one each of the replication forks moves to two sides of the replication bubble, and then it moves outward in both direction, that is bidirectional replication we say it has, and it starts to unwind and until it meets the DNA eventually. And if it's single frog, then the circulation process occurs twice, and then where we can see one will be old and one will be new strand. So yeah, that's all about the models of replication strand. I hope you all have understood. Thank you.